he is well-known international figure in research related to flash therapy. As a matter of fact, we just heard at least 1 or 2 quotations from his work. His PhD was focusing on brain and glioblastoma response to flash therapy. Also, he is currently leading a group of radiobiology team in his hospital and Iridium network in Belgium. His work research currently focuses on the mechanisms involved in the flash effect, along with preclinical studies aiming at translating flash radiotherapy to the clinic. His talk will be overview of flash therapy biology. Please, Dr. Bayer, can proceed.
Thank you very much for the nice introduction. So as you said, my talk today will be about the biology of flash radiation therapy. But exactly, I will try to answer to the question, what does flash radiation therapy do? So first of all, do we have a definition of flash radiation therapy? We know, and my colleagues Magdalena and Marco showed it pretty perfectly, that flash radiation therapy is delivered extremely fast. We are using ultra high dose rates, can be electrons, can be protons, X-rays, heavy ions. But the common denominator here is that it's ultra high dose rate. But what does that mean? Does it mean that it's only the ultra high dose rate that matters? But what is ultra high dose rate? What is a very high dose rate? What is the ultra high dose rate? Is there a threshold in time of the beam on time? Is there a threshold in energy or other beam parameters that are important? Those questions are still open. What we know on the biological side is that flash does not induce the classical radiation-induced toxicity patterns on the normal tissues. And on the other side, it has a very good anti-tumor efficacy compared to routinely used radiation therapy techniques. And that's what was called the flash effect. But is there a threshold in toxicity that can be accepted? Does it concern all normal tissues and all tumors? And I want to remind that observing the flash effect is an in vivo observation. So far, the flash effect hasn't been observed in vitro. We've seen observation of a normal tissue or normal cells preservation, anti-tumor efficacy in vitro, but both at the same time was not observed, actually, in my knowledge. So if I had to give a definition of flash radiation therapy, it would be a radiation therapy technique delivered at ultra high dose rate, but with specific beam parameters that is able to treat tumors without inducing drastic toxicity on the surrounding normal tissues. And I will start by talking about this normal tissue effect. Flash has not always been called flash. And if you look at publications that were published in the 50s up to the 80s, you can see that a radiation-induced skin toxicity has been described as dose rate dependent. And this was well showed by Inada and collaborators. And when they looked at skin reaction after the delivery of a dose, a high dose, up to 10 to the 9th, 2 times 10 to the 5th, gray per second, they observed that after they reach a certain threshold in instantaneous dose rate, the skin reaction was reduced compared to conventional dose rates. The same observations was done by Hendry in 1982, and Hendry looked at the necrosis dose 50 on the tail of rats, and he observed that when the dose rate in the pulse was above 10 to the 5th gray per second, this necrosis dose 50 was significantly increased, meaning that the toxicity induced by the irradiation was less than for conventional dose rate irradiation. And after this publication in 1982, and the publication you all know from 2014, from Vincent Favaudon, what really happened? In 2014, the terminology flash irradiation was used for the first time. What was new in 2014? Well, in 2014, Vincent Favaudon and collaborators showed that for a similar dose of 17 gray delivered to the thorax of mice, after conventional irradiation, you can observe, after 24 weeks post-irradiation, drastic fibrosis in the lung. But with the same dose, with flash irradiation, there was no occurrence, almost no occurrence of lung fibrosis. That was showed by Magdalena before. But what was very important in this paper is that for the first time, they decided to irradiate tumor-bearing animals and showed that flash irradiation was actually able to cure tumors in the lung of mice. So for the first time, they showed that ultra-high dose rate did not induce normal tissue toxicity, but still had a very good anti-tumor effect, with the possibility to increase the therapeutic index of radiation therapy. And that was called the flash effect. Back then, it was said that to observe the flash effect, you could use a mean dose rate of 40 gray per second or above, or a dose rate in the pulse above 1 million gray per second. And after that, many studies and many teams around the world show that flash irradiation using electrons, protons, photons, and it seems like I forgot heavy ions showed by Dr. Durante before. These teams showed that ultra-high dose rate was able to protect normal tissues from radiation-induced toxicity. That was shown on the mouse brain, on the mouse skin, bone, muscle, mouse lung, mouse gut, mouse hematopoiesis, but also on other models such as zebrafish embryos, on minipig skin. There were also vet clinical trials on dogs, so canine skin, vet clinical trials on cat skin, and a first study in 2019, the irradiation of a patient in a compassionate clinical trial performed in Lausanne. And all of these studies show that ultra-high dose rate electrons, protons, or photons induced low toxicity on normal tissues. If we look at which parameters shape normal tissue toxicity, you can find in any textbook that the total dose, the irradiated volume, the irradiation schedule, single dose or fractionation, the organ, and the individual variations will shape the normal tissue toxicity after irradiation. But when we compare conventional to flash irradiation, we usually do it at isodose. We use the same irradiated volumes. We compare similar irradiation schedules on the same organ, and we use genetically identical animals with a large sample size in order to avoid individual variations. So by any kind of textbooks, we shouldn't see any difference between conventional and flash radiation therapy. So how does flash induce less normal tissue toxicity? The only obvious different parameter between conventional and flash radiation therapy is the beam on time. So what happens really at the biology level? Is it something that happens at the cell level? Is it something that happens at the DNA level? Is it something that happens at the organ level? And all those endpoints were looked at by different teams and studies. First, people looked at DNA damage. If you don't observe toxicity on an organ, it probably means that there is less DNA damage after flash than after conventional irradiation. And that was observed. I cannot show you all the data that was produced about DNA damage, but that was observed by Levy and collaborators in 2020 on the gut, and they observed that there was less gamma H2AX and 53BP1 markers after flash irradiation compared to conventional irradiation in the gut after an isodose of 14 gray. What about cell death? Same thing. Favodon and collaborators in 2014 showed that there was less apoptosis, and more recently in 2020 showed that there was less senescence after flash irradiation in the lung compared to conventional irradiation. Then stem cells and progenitor depletions. We know that after irradiation, there is a stem cell depletion and progenitor cell depletion that leads to organ dysfunction. Well, we showed several times in the brain, but also in the gut, that after flash irradiation, there was a better pool preservation and proliferation of these stem cells and progenitors after a single dose here in the brain of 10 gray with flash compared to conventional irradiation. What about vascular damage? Well, vascular damage has also been investigated, and in all the studies, that was showed that flash irradiation induced less vascular damage investigated by preservation of endothelial cells and vascular integrity. Here, the results are obtained in the brain. Inflammatory response, same. Favreau and collaborators were the first to show it, but almost all the studies showed that flash induced less expression of pro-inflammatory molecules and less activation of innate immune cells. And about organ function, well, there I will show you results that I obtained on the brain, but it was also showed on the lung, on the gut, on the skin, on the muscles. For all those endpoints, the organ function preservation was showed after flash compared to conventional irradiation. So actually, you will just tell me flash does not do anything. And if you read the papers, it's always written flash radiation therapy group is similar to the non-irradiated control group, or flash radiation therapy does not induce blah, blah, blah. Well, that's what we observe. We have a control. The conventional is different from the control. Usually at the normal tissue toxicity level, we have an impairment of the function, for example, and the flash just looks like the control. Well, if we want to answer to the question, what does flash do? We will need to find something, a situation where we have the control, we have the conventional dose rate effect, and the flash dose rate effect is different from the conventional and from the control. So with my collaborator at UCI, we investigated the effect of flash irradiation on the complement cascade in the brain after irradiation. And first we showed, it was already showed before, but that when we irradiate with flash irradiation, we observe less astrogliosis in the brain compared to conventional irradiation. And we know that after irradiation, we observe an activation of the complement cascade, especially C1q protein, that leads to the production of anaphylatoxin, C3a, C5a, leading to chronic inflammation and astrogliosis in the brain. So we looked at the production of C1q, the activation of C1q after flash irradiation. And contrary to what we were expecting, since we did not observe any astrogliosis, we observed that there was an increase in C1q in the astrocytes after flash irradiation. Concerning C3, the anaphylatoxin, we observed that both flash and conventional irradiation were inducing the same amount of production of C3 anaphylatoxin in the astrocytes. So there was a discrepancy in the cascade after flash irradiation that we were not expecting. So we went further, and if you look at the activation of the complement cascade after irradiation, you have the production of C3a, C5a, leading to astrogliosis, but this depends to the activation of the TLR4 pathway and PI3K pathway. So we decided to look at TLR4. And we observed that actually after conventional irradiation, you have an increase in this TLR4 activation, both in the whole brain, but also specifically in the astrocytes, whereas after flash irradiation, this activation is not present, meaning that the difference we see in the astrogliosis is due to the absence of this TLR4 pathway activation. Nevertheless, flash induces something since we observed an increase in C1q and C3 activation in the brain after irradiation. So the big question we can ask about normal tissue toxicity is flash radiation therapy disrupting our knowledge of radiation-induced normal tissue toxicity? And the answer is a big yes. When we observe the difference between conventional and flash, we observed that isodose, same volume, same machine, same fractionation schedule, identical animals, but still we observe a difference that is not expected if we read the handbooks. We observed this different normal tissue response, meaning that there are other radiation delivery parameters that shape the normal tissue toxicity. And it's probably the time, the BMON time. And to investigate that, we need to choose the right models. Otherwise, we will never be able to answer to the question, what does flash do? And we have to choose them wisely, probably adapt them, or even develop new ones. But where does flash make a difference, really? Because I showed you that almost everywhere, there was no difference between flash and non-irradiated controls. Well, maybe this happens before the biological step, because after irradiation, there is a whole world that happens in a biological tissue before leading to what we call biochemical or biological steps. And this is physical, physiochemical, and chemical steps. And in 2019, we decided to investigate the effect of oxygen, and Professor Durante talked about that. And what we did is actually we decided to increase the oxygen tension in the brain before flash and conventional irradiation. We know that by increasing the oxygen tension, we would probably produce more damage with conventional, and we wanted to see if we could maybe change the outcome of flash irradiation or not. And what we observed is that when we treated the animals with flash, these animals were behaving just like the controls, non-irradiated controls, but when we increased the oxygen tension using carbogen breathing, we were losing the flash effect. In another experiment, we used antioxidant treatment on zebrafish to see if using antioxidant treatment could change the outcome of flash irradiation. And we showed that actually antioxidant treatments didn't change anything, and flash irradiation even preserved the development of zebrafish better than the group in which they were treated with conventional and antioxidants. And our conclusion in this article was that probably flash depletes oxygen in the biological tissues, and that protects the normal tissues. That led to probably dozens of papers studying the models. Is it possible? Is it not possible? And very recently, there was the first experiment actually measuring this depletion of oxygen in vivo that was published by Cao and collaborators in 2021. And the conclusion of this study is very interesting, saying that the magnitude of this change is much smaller than would be required to achieve global tissue hypoxia in normal tissues that are well oxygenated, meaning that the oxygen hypothesis, at least the depletion, does not stand. It cannot be the only explanation for flash radiation. And Professor Durante mentioned different possibilities. It could be the nature of the ROS produced. It could be the way they recombine, or other kinds of explanations. But at least we know now that the depletion of oxygen is not the main reason why we observe a flash effect. But what about the tumor? Well, the anti-tumor efficacy of flash radiation therapy was studied on different models. First on subcutaneous models, and it was shown that flash and conventional have the same effect on the tumor. Same on orthotopic xenografts, including human tumors that were injected in immunocompromised animals. Syngeneic orthotopic models were also investigated with mouse murine tumors that were injected in mice, so we can study the impact of the immune system, but also spontaneous tumors. One study on human tumor and studies on cat tumors and dog tumors. And all of these models showed an overall isoefficacy of flash and conventional radiation therapy. What about the tumor types that were studied? Well, murine glioblastoma was studied, murine lung carcinoma, murine ovarian cancer, murine lymphoma, murine head and neck cancer, feline squamous cell carcinoma, human skin lymphoma, and canine cancers. On all of these models, flash was shown to have an excellent efficacy, anti-tumor efficacy, with electrons, protons, and photons. So flash radiation therapy does the job. It can cure tumors. And you can read in papers, flash radiation therapy group is similar to the conventional radiation therapy group. Flash radiation therapy is efficacious to increase tumor growth delay. And what we observe is that we have the control. Conventional is efficacious against the tumor, and flash looks like the control. One big question we can ask is, are there situations where flash radiation therapy is different, and maybe even can the flash be worse than conventional radiation therapy. And in this context, we would have the control, conventional, and flash looking different. One study was performed to look at the differential effect of flash and conventional on the tumor microenvironment. And we know that the interaction of radiation therapy treatments with the tumor immune microenvironment is essential in the context of immunotherapy treatments. So this study by Kim and collaborators in 2021 showed that flash irradiation was able to preserve the blood vessel structure in a subcutaneous lung tumor model. And you can see here that after flash, we have a preservation of the blood vessel structure. They decided to investigate the level of tumor infiltrating lymphocytes that were in those tumors after conventional and flash irradiation. That was already described that after radiation therapy, conventional radiation therapy, we can observe an increase in tumor infiltrating lymphocytes in certain tumor types, and that was observed here with conventional. That was unfortunately not just a trend, not significantly different from the controls. But after flash, you can see that we really have an increase, significant increase in tumor infiltrating lymphocytes that was showed in this paper to be myosin light chain dependent, and meaning that flash could actually, in this context, be better in combination with immunotherapy, for example, or other kind of cancer treatments to treat the tumor. But unfortunately, it seems that all tumors do not respond equally to flash radiation therapy. That was a study performed by Shabi and collaborators in 2020, where they injected human T-cell acute lymphoblastic leukemia cells in mice. And in two of those models, M106 and M114, they showed that flash and conventional had at least a similar anti-tumor effect, sometimes even a bit better for flash on those tumor model, and it could delay the death of the animals. Unfortunately, for one model, you can see here that with conventional irradiation, all mice survived, whereas with flash irradiation, all mice died because of the tumor and at the same rate as non-irradiated animals, meaning that there might be tumor types for which flash irradiation does not provide the anti-tumor effect that we are expecting. So flash radiation therapy is efficacious against tumors, but we know that it works for high single doses in preclinical models, in vet trials, and in one patient. It also works for hyperfractionated radiation therapy regimen on one model of preclinical glioblastoma, and it works on small volumes. We also only looked at tumor growth delay so far. And there is a huge lack of studies about the anti-tumor efficacy of flash radiation therapy, especially about fractionated and hyperfractionated regimen. Only one study so far was performed on orthotopic models. The volume effect is very important. We are treating very small animals or very small fields. And if we want to translate that to humans, we might have to increase the volume of the irradiated field. Tumor heterogeneity hasn't been investigated either. Treatment combination, chemotherapy, immunotherapy, surgery, was not investigated. Treatment planning is not available so far, and local control was never investigated. And what about the differential mechanisms of action between flash and conventional? We still don't know anything about that. How does it work on the tumor? We have no answer. I want to remind everybody that tumor control is just as important, but even more important than normal tissue preservation. That's our first goal, to treat the tumor. And we need to perform those investigations to ensure a safe and efficacious clinical translation of flash radiation therapy. I want to go back to the pure biology now.
Just 1 minute, sorry. 1 minute to go.
And say that the flash effect opens a huge potential for cancer research, because flash radiation therapy protects the normal cells and kill the tumor cells. So we could actually use, in a very fundamental study, flash radiation therapy to study the intrinsic biological difference between normal cells and tumor cells in vivo, but maybe even in vitro. So flash radiation therapy is really a very powerful tool here. And my take-home message will be that there is a lot to do before understanding the flash effect, especially the mechanisms behind the absence of normal tissue toxicity, but also behind the anti-tumor effect, and to answer the question, what does flash do? Ultra-high dose rate irradiation is disrupting our knowledge of biological tissue interaction with ionizing radiations. We have to accept it and probably find new models and emit new hypotheses and solutions to study it. And we should not have any expectations on the results. And I showed you that sometimes we do not obtain, and that's science, the results that we are expecting. And I will conclude by saying that flash radiation therapy does not only have a clinical future, but it's also a very powerful tool for fundamental research and answer new questions in radiobiology. So this is my very small and fresh team in Antwerp, and I would like to thank the organizers of this meeting, my colleagues, and you for your attention today.